Hi, everybody. I'm glad to be here, and I want to thank the committee for this opportunity. So I'm a postdoctoral fellow that I started last year. So about GBA, so monoallelic mutation of GBA1 can increase the risk to develop Parkinson's disease, and among the mutation, the L44P and N370S can lead to misfolded protein that can lead to misfolded protein, while E326K decrease the interaction with LIM2. So this decreased the level of GCase in the lysosome. So one strategy to restore this defect is the use of a chaperone that will maintain the conformation of the GCase and increase the amount in the lysosome. So one chaperone that has been identified is the ambroxol. That is a FDA-approved mucolytic expectorant, and it has been shown to protect GCase against denaturation, and also it loses its interaction at acidic pH, and is under clinical trial. However, in vitro studies have shown that those 3 mutations can also have a decreased intrinsic GCase activity. So another strategy, and as you heard this morning, is the development of some activators, such as the BIA286156, previously known as LTI-291, and as you heard this morning, is under a clinical trial. So our hypothesis is the combination of both, so a chaperone and an activator, to synergistically increase GCase level and activity in dopaminergic neurons carrying the GBA variant, so L44P and E326K, and also the French variant W378G. But also, what we wanted to do is to try this strategy in context of defective lysosome. So as also you heard this morning, some GWAS association study identified several lysosomal genes as a risk factor to develop PD, such as CTSB and TMEM175. And we chose those two because the cathepsin B is involved in the maturation of the saposin C, which is the co-activator of GCase. And TMEM175 can regulate the pH of the lysosome. So to reduce the activity, the expression of those genes, we use CRISPR-I interference strategy in iPSCs to decrease the expression level. As you can see here by the qPCR, there are a decrease in the expression of CTSB, GBA1, and TMEM175. And we use DOX-inducible system to activate the transcription factor NGN2 to force the differentiation into dopaminergic neuron. And with the addition of the TMP, we can control the expression of the CRISPR interference system and induce it at the premature stage. So after 4 weeks, we look at the GCase activity using a live imaging probe, the LysoFQ, that emits fluorescence after cleavage by GCase. And this fluorescence increases over time, as you can see in the control curve, but not if we use the inhibitor CBE. So when we analyze the GCase activity for GBA interference and CTSB interference, we saw a decrease of the GCase activity and a mildly effect when we decrease TMEM175 expression. So we use this system to first evaluate the range of each drug and then see how the combination of those two can affect the GCase activity. So first, using a live imaging assay, we look at the effect of the chaperone on wild-type GCase with different concentrations, so 0, 1, 10, 30, and 50 micromolar. And we were surprised to see that after 5 days of treatment with 30 and 50 micromolar, so the pink and the yellow one that are done, we saw a significant decrease of the GCase compared to the control, so the blue one. And we saw a same trend when we used the activator, so a decrease when we use 30 and 50 micromolar doses compared to the control in blue. However, if we used the 4MU, which measured the GCase assay in the cell lysates, we didn't see this inhibition. But as our assay used live cell imaging afterwards, we decided to use nanomolar doses to evaluate the combination. So we used different range, 0, 1, 10, and 100 nanomolar of each of those chaperone and activator. And we use it first on the dopaminergic neuron with CTSB CRISPR interference. So we saw a reduction to 50% of the control level if you compare the white and the black bar. And when we compared with the activator alone, the chaperone alone, the combination 1-to-1 and the other combination, we saw a rescue to 80% of the control level for some combinations of, for example, the 100-to-100 ratio. And when we look at this 40-mem 175 CRISPR-I, which reduced to 80% of the control level. So when we add the activator alone, the chaperone alone, the ratio 1-to-1 and the other combination, we had a full rescue of the GCase activity with the 100-to-100 ratio, for example. So these are preliminary data we have to confirm, but it seems encouraging. And for those assays, we tried only for 24 hours treatment, so we want to increase the treatment duration to then also see if this can have an effect of some Parkinson's disease-related phenotypes, such as alpha-synuclein level, for example. And of course, at the end, we want to try this on the patient lines also. So I want to thank again the committee for this opportunity. I want to thank the Edward Fond Lab and the Zip Gardner Lab and all the members, the Silverstein Foundation for the fellowship, and everyone for the attention. Thank you.